Dr. Church, why did you choose to enter the field of genetics and synthetic biology?
So I had an early interest in all kinds of science, and it seemed like biology was really complicated and interesting. And I had a kind of a choice between analytic and synthetic, uh, anything, chemistry, biology, whatever. And analytic is observational and reductionistic, while synthetic is um, more constructive, and it gets us closer to where we want to go in terms of fixing medical environmental problems. And genetics is a nice partner for synthetic biology. It allows us to very easily read and write and program all kinds of complex biological systems, ranging from atoms to ecosystems.
Who would you say were your greatest mentors you met along the way?
Yeah, well, I mean, I was fortunate that a lot of, well, also just by proximity, you tend to either think your own mentors are the best mentors, or you've had some problem with them. But I'm lucky enough that all my mentors have been terrific, and so in my, I mention them as often as I can. Even in my thesis, I mentioned my PhD thesis. I mentioned my high school mentors who were Creighton Bedford and Don Snyder. They weren't on my thesis topic. They were on math and photography, but they made just a huge impact. And I have visited them recently. Then by college, I was working with Sung Ho Kim. He was really the first one that I did my first five scientific papers with him on crystallography, and that got me thinking about nucleic acids, because we happened to be doing crystallography on one of the first nucleic acid folded molecules. And he was just a great influence, and he's continued to, he was there when I got into the National Academy of Sciences just fairly recently, relative to how long we've been working together for 50 years off and on. And then for my PhD and postdoc, Wally Gilbert and Gail Martin, these were all exemplary, um, for being on the cutting edge of chemistry and biology.
What inspired you to initiate the Human Genome Project and the Personal Genome Project?
So I had a big grant proposal I was submitting, and I realized as I was doing it that we were inventing a new way to sequence DNA, which was approaching so fast. I mean, an exponential uh, approach that it seemed like we were likely to be reducing the cost of sequencing maybe a millionfold, maybe, as it turned out, 20 millionfold. And it was a good time to start dealing with, important topics and to my dismay, I learned about human subjects research for the first time, and found a bunch of things that I felt were not as ethical as they should be. These are sort of the ethics standards, and they were saying things like, well, if you learn something um, um, extremely informative about somebody's health, you can't communicate it with them, and you also can't share the data, even if the patient wants you to share the data. And so I explicitly figured out how to do that in the Personal Genome Project, and wanted to use that as an example for other people that were uncertain as to how to deal with these kind of ethical issues, that so they could share their cells, share their DNA sequences, without imposing restrictions if they don't want them. If they want their particular genetics and diseases to be front of the line, then they should be free to do that. It's kind of like,
You work both in wet lab research and the biotech startup industry. How do you balance the demands of both jobs?
first of all, they're not that demanding. They're a lot of fun. Second of all, they reinforce one another, and they synergize in such a way that I basically gain time by doing a good job at both of them. Essentially my contract at Harvard and MIT requires that I only spend 20% of my time outside of university activities, which is not hard to restrain myself to just 20%, because you can be pretty efficient when you put your mind to it on things out and inside the university. The goal is not so much to be efficient as just to try to help out students and postdocs and get research done.
Do you think the US needs in terms to stay competitive in science and medicine?
I would say that, one small possibility is that grant funding tends to focus on kind of two extremes. One is like the extreme basic science, where it's just like you don't have to justify, it's just basic, and then at the other extreme is like, oh, it has to apply to human health, period, and in between there's all kinds of what I like to, you think of, there's basic and applied science, but there's also basic applied science, or there's pure science, sometimes to be distinguished from applied, but then there's pure engineering, and I think those are quite interesting, where you have radical, especially if you focus on radical and fundamental technologies. By radical, I mean something that is completely non-obvious, but could change a lot of things. And fundamental is something that applies to many different fields, like sequencing and synthesis, reading, writing, and editing DNA allow you to do all kinds of, it's sort of organism agnostic, it's theory agnostic, etc. It's just very general tools.
AI has been a big technology recently, and it continues to grow. Where do you see the use of AI in genetics and synthetic biology?
Genetics and synthetic biology has a rich history of using computational methods in general, but we're entering a new phase. My lab's published, 10 or so papers on applying it mainly to protein, nucleic acid, and cell design. So design is different from, predicting three-dimensional structure. You're trying to design for function, which very often isn't merely a matter of finding its 3D structure. In fact, sometimes you can make radical departures in function without knowing the 3D structure, and without knowing the 3D structures, all things it's interacting with. So, for example, we've used machine learning on the gene therapy delivery mechanisms, like viral capsids, where you're trying to get it to go to position X in the body and not position Y, and you don't really know all the possible things it could be interacting with in exquisite detail, but you can nevertheless do that. And we've done this, for example, BCAP1 at Dino Therapeutics is amazingly targeted for the brain. So yeah, I think there's going to be just no end of it. But in a way, it's just an extension of previous successes in prediction and design in biology.
What are you currently working on in the lab? What are some of your biggest projects right now?
It's a complicated lab. I would say maybe 80% of what we do is synthetic biology, and the other 20% is analytic, but they're highly entangled. The analytic was mostly focused on in situ methods, or sometimes called spatial omics, spatial multi-omics, but we do it by microscopy rather than other methods. So we directly observe things in situ. That's the analytic side. On the synthetic side, we're doing things like what we just recently showed, that we could get resistance to all viruses by a simple recoding scheme where we swap two codons from serine to leucine, and then we think that could apply to pretty much any organism that has a virus problem, agricultural, endangered species, human, so forth. We're doing we're not only making organs for transplants from pigs to humans, but making them enhanced so they won't succumb to the diseases that got rid of the first organ. Resistance to pathogens, resistance to cancer, senescence, and so on. I teach a course called How to Grow Almost Anything at Harvard and MIT, and actually worldwide through online. But we take that seriously. You know, how can we? Because growing things is probably going to be less expensive and ultimately more atomically precise than just about any other engineering we can do. So those are some samples, but I could go on. I won't. I could.
What has being a scientist taught you?
I think I discovered somewhere along the line that I was a scientist slash engineer. Um, and and one of the cool things about being both is it makes it hard to completely fail. And you can still fail it. So if you like make this big engineering plan, you know, you got it all, it's very complicated, lots of moving parts, and it works first time, then you're a competent, capable engineer. On the other hand, if it fails and it's a mystery why it failed because you carefully planned it, then you made a discovery probably, and in the process of following up that discovery, then you become a good scientist for a while until you move on to the next thing.
What advice do you have for scientists who are just starting their career in science?
Well, the first thing before I give any advice, I have to say ignore, don't trust advice, especially if it's one size fits all. So that's the disclaimer and advice. And very often there'll be a lot of pressure to conform. I think if you're in the not the one size fits all, but if you are in the kind of place that I'm in, then you want to be bold and step away from the crowd, because they're either wrong or there are so many of them that there's no space to grow. You don't have to. I'm just saying that's one way of succeeding. There are many ways to succeed in science. Also, I think what happens is sometimes people think of science as being less social than the social sciences, and it's almost the opposite. I find a lot of my professor friends in the social sciences work pretty much by themselves with very few students, and they're writing their next book, which requires focus. While a lot of my medical research friends are running labs with 20 or more people in them. So there's a lot to be said for communication, clear communication, constant but concise. Be brief about it.
Everyone receives mentorship from professionals when they are starting their career in science, but what qualities do you believe make a good mentor in science?
I mean, I can mention things that I appreciated among my mentors, and and hopefully my students will improve on whatever failings I had as a mentor. So I think patience is one, humility, sympathy, creativity, working with people to encourage their creativity, not just your own. And also it's important to, there's a lot at stake when you're interviewing somebody. You're kind of not only figuring out if they're a good match. You don't want to, it's not good for them or the lab to bring in bad matches. But also you're kind of setting the tone. You're sort of setting up, what your principles are for the laboratory, and that can help them decide whether it's a good fit. But it can also start the conversation about, what they should be doing to conduct themselves responsibly.